We're talking about capillaries, and as we're talking about capillaries, I tried to make the point that even though arteries and veins are great, they're a perfect way to get some blood from the heart to a part of your body. They are not sufficient for delivering whatever is in the blood. Any individual substance, whether it's a little bit of protein or a little bit of sugar or a little bit of oxygen, if it's going to actually be delivered directly to the cell that needs it, it has to be delivered through these things called capillaries. And we stopped here when we were talking about how capillaries are very thin and they leak a little bit. And the reason that they leak a little bit is they need to. Let's look at that. Here we've got a typical capillary. Your body's got 3, or you could say 4 different types of capillaries. The capillaries in the brain are this type of capillary, but they got extra covering around them called the blood-brain barrier. Almost everywhere in your body, you've got these typical capillaries. We're going to be learning about a type of capillary where the cells here have got extra little holes through them. So they're extra leaky. Those capillaries are called fenestrated capillaries. And in the liver, there are capillaries that are called sinusoids, and they're just ridiculously leaky. But for the rest of this lecture, we're just talking about a typical capillary. Now, if you are a molecule of glucose in here, you are dissolved in the watery part of the blood, and there's a cell sitting right here that would love to have that glucose. However, glucose dissolves well in water. It is hydrophilic. And since it is hydrophilic, it cannot go across the cell membrane, right? Remember that from endocrine, from a bunch of places. Now, molecules like oxygen that are hydrophobic, they don't dissolve well in water, but they do dissolve in the phospholipid bilayer, and they'll just sneak their way around the cell membrane and go to the next cell. So this particular talk is about how has the body designed this system so that all of the goodies that are dissolved in the watery part of your blood that are hydrophilic, how do they get to the cells where they need to go? And the answer is they go through these little holes. Now, these little holes, these intercellular clefts, they're tiny little holes, and it's not like having a great big old giant hole in a garden hose. It's more like, you know those garden hoses? There are garden hoses, and they're made out of stuff that feels kind of foamy. And if you turn the water pressure on, you never get it springing a leak, like, psst. No, it doesn't do that. But it just kind of gets wet, and then it just kind of drops of water come off. That's kind of what these typical capillaries are like. So here we've got our capillary bed, and there are cells in there and cells in there that they want the glucose or the insulin that is dissolved in the watery part of the blood. Here's what's going to happen. At the beginning of the capillary bed, the blood pressure is pretty high, and so it creates hydrostatic pressure that's pushing against the wall of the capillary. Kind of like when you turn on that leaky garden hose and the water pressure is pressing against the garden hose and it starts to leak. That leaky stuff is going to splash across the cells. All the cells are living in the white part of this particular image. So that water with little molecules that dissolve in the water will splash across here. And as they're splashing across, they'll go, hey, as long as I'm here, would you like some glucose? And then the cell will say, sure, I'll take a glucose, but hey, could you get rid of this CO2 for me? And then the water will say, sure, I'll pick that up. And it goes splashing across. And then at the end, that water, now it doesn't have glucose in it, doesn't have amino acids in it, doesn't have insulin in it anymore. It's picked up all this trash. Now that water, it jumps back on board into the end of the capillary, and then it gets heading back to the heart and on its way, right? Now, the question you should be asking me is, but Tidal, how does the water know to jump out here and to jump back in here? And the answer is hydrostatic pressure and osmosis. Right? So let's review briefly the way blood usually flows. Blood usually will go from one of the ventricles of the heart into a big artery like the aorta. then smaller arteries that have got names, and then some arteries that don't even have names. Then it'll go into an arteriole. After that, it'll go here through a capillary bed. Then it gets picked up by tiny little veins called venules. Then it'll go into a vein, and then veins with names, and then the vena cava, and then back to the atrium, and then we'll go to a ventricle, and round it goes. So, control of capillary bed perfusion. We're going to be talking about the way the watery part of the blood is going to leave the capillary at the beginning, splash across all this yellow stuff here, and jump back in at the end. But before we do talk about that, let's talk about the control of capillary bed perfusion. Let's imagine that you're way ahead of me and you actually have this picture in your head that you not only have arteries and veins, but you've got this really delicate network of little tiny blood vessels that are carrying blood and delivering stuff to the cells. If you do have that picture, you probably also have the mental picture that there's always some blood running through them. The truth is, there's not. Particularly while we're just sitting here, I know I'm just sitting here, in our skeletal muscle, the capillaries there are closed most of the time. They're just closed. There's no blood going through them. Now you might think, oh, aren't those skeletal muscle cells that live in those little yellow spaces, aren't they sad? No, those skeletal muscle cells right now, they're not doing anything. So they're cool if there's no blood going through it. The neat thing about capillary bed perfusion is that one way of controlling whether there's blood running through here or not is going to be the dictates of the cells that live there. So let's say that there are some skeletal muscle cells in there, and they're like, you know what? I'm starting to get hungry. I'm starting to need a little bit of oxygen. Let's have some blood run through here. They will send out molecules, and those molecules will cause these little cells here that kind of look like turbines. It's going to cause them to relax. And when these cells relax, blood goes flowing through, and the cells that are living there are skeletal muscle cells. They get all the glucose and amino acids they want. They get oxygen. They give away all their trash. And when all of that is done, they go, oh, okay, I don't need blood anymore for a while. So while you're just sitting there quietly, any given capillary bed in your leg muscles is staying closed for about 60 seconds, and then it'll open for 10 or 15 seconds, and then it'll stay closed for 60 seconds back and forth. So most of the time, most of your capillary beds are closed. And by the way, that's a good thing because the human body does not have enough blood to have blood go through all of our capillary beds all at the same time. We don't have enough blood pressure. It would be kind of like, let's imagine you live in an apartment building, and on any given morning, pretty much everyone takes a shower. 4 out of 5 houses take a shower at the same time, and you always get good water pressure. But let's imagine on one day, just for the heck of it, everyone turned on their shower, turned on their dishwasher, started a load of laundry, turned on the sink, and flushed the toilet all at the same time, okay? You wouldn't have any blood pressure, any water pressure. Well, that's because there's only so much water to go around. That's the same way it is in the human body. We don't actually have enough blood to put blood through all of our capillaries all at the same time. If all of your capillary beds open up all at the same time, then your blood pressure crashes to almost nothing, and it'll stop your heart from working right, and that can kill you. So what controls capillary bed perfusion? It's going to be the cells that live in these white spaces, whatever they are. When they have used up their oxygen, they have made a whole bunch of trash called carbon dioxide, CO2. And when carbon dioxide dissolves in water, it creates acid. And an acidic environment and lots of CO2 is going to cause these cells to relax. And when these cells relax, then blood goes through the capillary bed. So it's kind of like the skeletal muscle cells are ordering pizza whenever they want it. And when they're not hungry, they tell the pizza delivery guy, don't come. So let's get back to the idea that there actually is water leaving the beginning of a capillary bed and then jumping back on board at the end of the capillary bed. And I asked you that, if it made you wonder, how does water know where to leave and where to jump back in? Well, of course, water doesn't know where it is. But the reason it happens is because we have got a tug of war between the force of osmosis and the force of hydrostatic pressure. Remember hydrostatic pressure? That is what runs filtration. This little spaces in between, the little spaces in between the simple squamous epithelial cells are super small. They will allow water and very small molecules that are dissolved in water, they will allow them to leave if the pressure inside is greater than the osmotic pressure asking the water not to leave. Remember, water gets drawn towards wherever there's lots of solute. Water gets pushed away by hydrostatic pressure. So they're fighting each other, right? So hydrostatic pressure is going, I want the water to leave. And osmotic pressure is saying, no, don't you leave, okay? So they're fighting against each other. Well, it happens that blood pressure at the beginning of a capillary bed is relatively high. It's significant. It's 35 millimeters of mercury. And at the end, it's very low. We'll get to that in a second. At the beginning, the osmotic pressure, the pressure that's saying, don't leave me, water, that osmotic pressure, that is being generated by the red blood cells that are in there and the platelets, and a lot by albumin. Remember I said albumin was there for its osmotic pressure principle? But you'll notice that 35 is greater than 24, so out wins, okay? Hydrostatic pressure and osmotic pressure are fighting each other. Hydrostatic pressure wins, and we're going to get some water leaving at the beginning of a capillary, splashing across the cells, delivering its glucose along the way. Now, at the end, what happens? The hydrostatic pressure has dropped way down. Hydrostatic pressure drops because of the network nature of the capillary bed. But now the hydrostatic pressure has dropped, and the osmotic pressure, well, it didn't go anywhere. There's still as many red blood cells, there's still as much albumin inside as there was before. So now inward wins. So at the beginning, outward won the tug of war. At the end, inward won the tug of war. So that is how things leave and enter. Let me summarize it by saying water and small molecules that are dissolved in the water, like glucose, amino acids, insulin, they're going to leave at the beginning of a capillary bed because, you might want to write this down, hydrostatic pressure is greater than osmotic pressure. Whereas they're going to jump back in at the end of the capillary because hydrostatic pressure is much lower than osmotic pressure. Elegant, and another reason why you needed to know osmosis. Now, the pulmonary capillaries, they are different. They are still continuous capillaries, so they're a typical capillary. What is different about them? What's different is the blood pressure in them is much lower. So since the blood pressure in them is so much lower, that means that almost no water leaves at the beginning and all of it comes back in at the end. All right, we'll pick up there with blood pressure on the next video.